journals, and it was a challenge to find a paper without my advisor's name on it, so this one. Second, the paper looked at the combination of physical activity and cognitive activity on the brain in humans, not in animals like most of the time. And third, it involved a genetic aspect, specifically dopamine-related gene polymorphism that I found myself very interested in. So to give you a little bit of background, age-related cognitive decline and diseases such as Alzheimer's and other dementias impose a huge burden on patients, relatives, and public health system. Especially the problem becomes very important as aging population grows. So today we have around 5.4 million of people who have Alzheimer's, and by the year 2030, this number will reach 7.7 million. So what can be done in order to offset the cognitive deterioration and reduce the incidence of age-related brain disorders? Some studies suggest that physical activity and cognitive enrichment might help. Both of those factors individually induce reorganization of cortical and subcortical structures as shown in humans and only in animals, but only a few studies in humans were done that combine the two and evaluate the effect. So let's look at the exercise-induced brain plasticity first. In this study, conducted by Erickson and others, there were two groups. One that exercised aerobically and the other one that was engaged in stretching and toning for control purposes. So over the period of 1 year, the control group's hippocampus volume decreased, and the stretching exercising group hippocampal volume increased. So this paper showed, this study showed that exercise can increase hippocampal volume in older individuals. So all the studies I'm going to talk about were conducted in older individuals. So that's important.
So in this study, they do any like memory tests or anything like that to look at like a behavioral effect?
Yes, it was correlated. The memory was improved as well, and it was correlated with the increase of hippocampus. Another example of impact of exercise on the brain was the study. There's also the study by Kalkomba and others, where they showed the increase in gray and white matter in prefrontal cortices, so frontal cortex. So here we see the map of exercise minus control. So, that's how they show it. Learning and other cognitive enrichment seem to have an impact on brain plasticity as well. So in this study, 8 weeks of memory training increased the cortical thickness in different brain areas, including insula that is shown in this picture. So we have control group and training group, also older adults, at time point 1 and time point 2. So in control group, the cortical thickness decreases over time, and the training group, the thickness increases.
Is the insula related to Alzheimer's?
I don't think so. But
So what is the importance of this increase in insula?
I just wanted to show that there is a reorganization of the brain structures that are triggered by such things as cognitive training, memory training.
So in the study, did they not look in other places, or is this?
There were other places. It just was a good picture. So there were other places that were also showed an increase in cortical thickness, but I just did not show that. So another example of learning-induced brain plasticity would be the increase in hippocampus, in gray matter of hippocampus in the older adults who learned 3 volts juggling. So it was a transient increase, but still, there's something going on that is triggered by the training. the cognition. So, what about the combination of physical exercise and cognitive stimulation? So studies in animals shows that those effects are additive. Locomotion stimulates the precursor cells, while learning and other enrichment promotes the newborn neuron survival. And that's what's shown in this study. There is a gradual increase of neurons born and neurons survived as we move from the control group to the combination of activities such as running and enriched environment. So here is this control group. This is enriched environment by itself. So enriched environment is like different toys for rats and stuff like that, mazes, and running wheel group, and a combination of two. So it shows the additive effect, and I wish there were studies like that in humans, but I didn't find any. So to summarize, there is a positive influence of physical activity and cognitive activity on the brain at the neuronal level that is shown in animals, as well as an increase in cortical and subcortical structures that's shown in humans. What about the influence of those factors on the communication between different brain regions? So one of the great techniques that is good for characterizing the nature of interactions between different brain regions is functional connectivity analysis. And this is the type of temporal coherence analysis between the remote regions. And one of the large-scale brain networks that is evaluated by this type of analysis is default mode network, DMN. So it's going to be reported in the paper. It includes medial prefrontal cortex, posterior cingulate cortex, inferior parietal lobule, and hippocampus. DMN is associated with the reflective activity and self-referential mental processes. Also, aging is related to the decreased DMN overall activation. One study by Voss and others evaluated the influence of exercise on the functional connectivity within the DMN. And also they had one group that engaged in non-aerobic exercise and in aerobic exercise. So after a year of walking, they showed that there is an increase in functional connectivity within DMN in the aerobic exercising group. So that's
how do they measure functional connectivity?
Functional connectivity is measured by fMRI. So.
And what in particular do they look for in the fMRI?
The coherence between the activation of different networks.
Different regions.
Different regions, yeah. So, finally, I wanted to ask what neurotransmitter systems are implemented in brain aging, and can activity, physical or cognitive, modulate those systems? So dopamine, without a doubt, plays a huge role in cognition, and there are individual differences in DA functioning that is linked to cognitive performance. Also, the dopaminergic system serves as a mediator of age-related decline in executive functioning. Important to note is single nucleotide polymorphism of COMT enzyme, which is an enzyme that breaks down dopamine, can be attributed to some of those cognitive performance differences. Better performance in MET homozygous carriers were shown comparing to the heterozygous MET-val carriers. As for the impact of cognitive training, it has been shown that there are changes in cortical dopamine D1 receptor binding following 5 weeks of memory